Good afternoon, everybody. It's a pleasure for me to be hosting the Showcasting Discoveries at the Josep Carreras Leukemia Research Institute Open Lectures of its group leaders. Today, we have the honor to have with us Dr. Fumiichiro Yamamoto, who will be discussing about ABO blood groups from basics to molecular genetics, genomics, enzymology, evolution, and disease. But before we start, let me provide you a bit of background about Dr. Yamamoto, who's one of our group leaders and is in particular the group leader of the Immunohematology and Glycobiology group. Dr. Yamamoto received a PhD in developmental and cellular biology from Osaka City University in Osaka, Japan. After postdoctoral work at the State University of New York at Stony Brook in New York, he became the head of the molecular biology laboratory at the Biomembrane Institute in Seattle. Later, he joined the Brigham Institute La Jolla in California. After holding the Chair of Excellence Pierre de Fermat, Paul Sabatier University, Toulouse, France, he was recruited to the Institute of Medicina Preventiva y Personalitzada del Càncer in Barcelona. In 2015, Dr. Yamamoto's research group joined the Josep Carreras Leukemia Research Institute. I'd like to mention that Dr. Yamamoto has received several international recognitions, including the Doctor Honoris Causa from the French Republic, from the Paul Sabatier University, and the Karl Landsteiner Memorial Award for his elucidation of the molecular genetic basis of the ABO blood group system. So the stage is yours, Dr. Yamamoto, but before we start, let me remind the audience that please keep your microphones muted and during the whole talk, and then send your questions through the chat where I will be reading them. And please make them. The best student that asks a question will get a coffee from me. So Yamamoto, Dr. Yamamoto, the stage is yours, as I said.
Thank you, Mariana, for the introduction. Good afternoon. My name is Fumiichiro Yamamoto. Can you see? Can I? I cannot change the slide to the next one.
Click this, start to the screen, and then the arrow.
Okay. Sorry, it might take longer than I assume because of the technical problem. There are 6 learning objectives in this talk. They are ABO basics, molecular genetics, genomics, disease association, COVID-19 association, and ABO GBGT1 crosstalk. My laboratory has been studying cancer, developing molecular biological techniques, and investigating ABO. We have been studying the ABO blood group system, ABO genes, A and B glycosyl transferases, and A and B oligosaccharides, employing a variety of scientific disciplines, including molecular genetics and immunohematology. Since we moved to Spain in 2009, we have expanded our research interest to include other members of the ABO gene family of alpha-1,3 GalNAc transferases. We have been especially interested in Forstmann. Because we are interested in anything relating to ABO, we call our study Glycobiology. History. In 1492, 3 young men were sacrificed, and their blood was orally given to the Pope, who was in coma. But he died without beneficial effect. In 1616, William Harvey discovered blood circulation inside the body, and the idea came out that blood should be transfused into the circulation. Jean-Baptiste Denis and Richard Rohr performed the animal to human blood transfusion in France and Britain, respectively. But because of the high risk, blood transfusion was prohibited in these two countries. In 1818, James Brundle, obstetrician, performed the first successful human-to-human blood transfusion. He later reported that blood transfusion was beneficial in 5 out of 10 cases. Patients were women who lost a large quantity of blood during baby delivery, but he could save 5 of them. In 1900, Karl Landsteiner discovered the ABO blood group polymorphism. He isolated cellular component of blood, red blood cells mainly, and liquid component of blood, serum, and from himself and his colleagues, and mixing combinations. When they are derived from the same individuals, nothing happened, as shown with a minus symbol, but in some combinations, he observed the agglutination of red blood cells, as shown with the plus symbols. If this happened within the body, it's troublesome. The agglutinated red blood cells may be lysed because of the functions of complement, and hemoglobins are released into the bloodstream. If kidneys, which filtrate blood, fail to function normally, the recipient may die. Therefore, performing the test, this agglutination test, before transfusion, avoiding the combinations resulting in red blood cell agglutination, and transfusing only the matched blood became the principles for safe blood transfusion. Because of this discovery, Landsteiner received the Nobel Prize. Another important finding from this study is you can see 3 different patterns in the table. The fourth pattern was discovered the following year, and the individuals could be categorized into groups based on the agglutination patterns. So A, B, O, and AB blood groups. In order to explain this phenomenon, Landsteiner postulated 2 kinds of antigens, later named as A and B antigens, and the antibodies against these antigens in the individuals who do not express these antigens. These antibodies were called natural antibodies because they were present in healthy individuals who have never received blood transfusion. Group A individuals have A antigen expressed on red blood cells and contain anti-B antibody in the serum. B individual express B antigens on red blood cells and contain anti-A antibodies. AB individuals express both A and B antigens. On the other hand, O individuals don't express these antigens but contain both anti-A and anti-B antibodies. This AB polymorphism was soon found to be the hereditary trait that follows the Mendelian law of inheritance. Although both antigens and antibodies are involved, this polymorphism is specified by alleles at a single genetic locus ABO. In order to explain the mode of inheritance, in 1924, Bernstein proposed the one genetic locus, 3 alleles model. He postulated that the A and B alleles are codominant against recessive O allele. Because of the combination, there are 6 ABO genotypes, resulting in 4 phenotypes. The ABO blood group distribution varies depending on races, countries, and geographical location. As you can see in this world map, group of individuals prevail in the American continent. Actually, before the colonization, all the people living there are supposed to have group O. Abiotyping can be done by two ways. Forward typing detects the antigens on red blood cells, and reverse typing detects antibodies in serum. Most of the samples show very strong agglutination or no agglutination at all. However, there are some samples which showed weak agglutination, and these were categorized into A and B subgroups. A2 allele or A2 phenotype is relatively common in the Caucasian population, but all the other A and B subgroups are very, very rare. When we say group AB, that is in trans situation. A allele comes from one parent, and B allele comes from the other parent. But there are some cases, although very rare, which seems to specify one gene encode the expression of both A and B antigens. When the expression is weak A and weaker B, that gene was called cis-AB. When the gene specifies the strong expression of B antigens and weak expression of A antigens, the allele was called B parenthesis A. A and B glycan antigen, these antigens are not protein antigens, but glycans. The antigen structure is galactose linked with fructose by alpha-1 to linkage, and also linked with agarnac by alpha-1,3 linkage. P antigen is similar to A antigen, but in place of agarnac, galactose is linked to galactose. The difference between agarnac and galactose is the side group in the second carbon position of the sugar ring, which is an acetyl group in agarnac and hydroxy group in galactose. These glycans can be parts of glycoproteins or glycolipid, or they may also exist as oligosaccharides. And on the cell surface, glycan face outside of the cells. A single group A1 red blood cells carry 1 million A antigens, whereas group B red blood cells carry more than 500,000 B antigens. And red blood cells of subgroup carry much less antigens. A and B antigens were initially identified on red blood cells, but later found to be expressed on other types of cells, including epithelial cells of gastrointestinal and respiratory tract, as well as endothelial cells that line blood vessels. Because of this, the avian matching is also crucial in the transplantation of cells, tissue, and organs, in addition to blood transfusion. A and B antigens can be expressed on glycoproteins in secretion. Saliva, seminal fluid are important biological evidence in addition to blood, skin, and hair at the criminal scene. Actually, ABO polymorphism is the most important biological evidence to exclude innocent people from potential suspect before DNA typing became popular. The International Society of Blood Transfusion recognized 43 blood group systems of red blood cells, and 7 of them are polymorphism of glycosylation, including ABO and Forstmann. And these glycan antigens may also be expressed on other types of cells, because in humans there are more than 200 different types of cells. A large number of glycosylation polymorphisms may exist. Group O individuals don't express A or B antigens, but express H substance that is characterized by a galactose linked to its fructose by alpha-1,2 linkage. Based on the structure of H substance and A and B antigens, a hypothesis was proposed on the biosynthesis of A and B antigens from the common precursor H substance by the transfer of a GalNAc or galactose by A and B transferases encoded by A and B alleles at the abiogenetic locus. It was in 1959. In 1990, 90 years after the discovery of the ABO blood groups by Landsteiner, in collaboration with Henrik Clausen and Seiichiro Hakumori, my research team cloned human A transferase cDNA and later B and O allelic cDNAs and determined their nucleotide and amino acid sequences. By correlating their sequence differences with the expression of A and B antigens, we demonstrate the hypothesis on the biosynthesis, which became the central dogma of ABO. Namely, A and B alleles encode A and B transferase, which synthesize A and B oligosaccharide antigens. We found 4 amino acid substitutions between A and B transferases. We also identified a single nucleotide deletion 261 deletion G relatively close to the N-terminus of the coding sequence in O alleles, elucidating the molecular genetic basis of the ABO system. We became the first who succeeded in ABO genotyping, discriminating A genotype from AO genotype and BB genotype from BO genotype, which was impossible by the immunological method. We moved on to identify a single nucleotide deletion at the C-terminus of the A transferase coding sequence in A2 allele, missense mutations in A3AX, A3 subgroup alleles. We showed that cis-AB and B parenthesis A alleles specifying the expression of both A and B antigens by single alleles encode AB transferase chimeras. In addition to O allele with a single nucleotide deletion, we also identified O allele with missense mutation. We set up the experimental system to analyze the functionality of the mutation. We used the DNA transfection of HeLa cells. We first showed that HeLa cells express H substance. When A or B transferase are produced inside the cells by DNA transfection, that can transfer GalNAc or galactose to H substance, making A and B antigen respectively, which could be detected by the antibodies. Using this simple system, we first showed that A1 construct and B construct induce the expression of A and B antigens respectively. We then introduced O allele specific single nucleotide deletion or amino acid substitution into A1 construct. No expression of an antigen was observed, meaning that these mutations are inactivating mutations. We then introduced A2 allele or A3 allele specific mutation, and in these cases, antigen expression was still observed, but decreased. There are 4 amino acid substitutions between A and B transferases. We made 14 AB transferase chimeras having amino acid of either A or B transferase at those 4 positions and determine the sugar specificity. What we found was amino acid at codon 176 are not important at all. Codons 235, that may be influential, but a little bit. The most important are amino acid at codon 266 and 268. They are leucine and glycine in A transferase and methionine and alanine in B transferase. We characterized only the first 12 ABO alleles. It is surprising that only with the sequence data from 12 alleles, we could determine the ABO phenotype with 99.9% accuracy. Later, many more ABO alleles were characterized by other scientists, and even the database was produced. 250 alleles were deposited in the database. 4 amino acid substitutions were conserved in the majority of the B alleles. The single nucleotide deletion was also observed in the majority of O alleles. However, other mutations were also identified, including those in the promoter and enhancer region. But thanks to the recent advancement in the next generation sequence, many, maybe millions of human genomes have been sequenced, and many single nucleotide variations have been identified in the human gene. Here shows different mutations in different colors. Actually, out of 1,065 bases in the coding region, at 348 positions, already single nucleotide variations have been identified, and this number is rapidly increasing. The APO gene spans over 25 kilobases in the genome, and more SNP variations have been identified in the non-coding region. If alleles are defined as genes occupying the same genetic locus, but possessing the different sequence, the number of the alleles may amount to 1 million, as opposed to only 3 proposed by Bernstein. In 2012, predicting this situation, we proposed a nomenclature of alleles based on the assortment of SNPs. ABO has become one of the genetic loci most studied for human polymorphism. The repertoire of ABO polymorphism varies depending on the species. We characterize the partial nucleotide and amino acid sequence of primate ABO genes from several species. We found that leucine and glycine and methionine and alanine are conserved in primate A and B genes respectively, meaning that these amino acids are important in discriminating the sugar of agarnac and galactose. We found mouse ABO gene encode cis-AB transferase. Pig has AO system. We clone the A transferase cDNA, and we show that pig or allele lacks a majority of the structure gene, as opposed to human or alleles, which have a very minor change of single nucleotide deletion or amino acid substitutions. Actually, now many species are known to possess the ABO genes. A lot of useful information is now available from databases such as GenBank and Ensemble. For example, gene organization, exon boundary, and SNPs. We studied the evolution of the ABO gene. We made a variety of human A transferase expression construct having amino acid substitutions around codons 266 to 268 and also 263 to 268 in some, possessing the amino acid found in different species. And by DNA transfection, we characterize the specificity of this construct and made this code table. Before we knew that the most important amino acids are codons around Gs 266 to 268, but there may be additional mutations which may affect the specificity. But still we hope to obtain some important insight. We extracted many ABO genes from the database and made phylogenetic trees. And here shows one. Circles and triangles shows potentially full and partial genes. And we assign the sugar specificity and color coded. You can see different patterns. But actually, this phylogenetic tree shows several species that possess multiple non-allelic ABO gene sequence, and several of them have both A and B gene sequence. This is quite different from humans and primates, where ABO genes are alleles occupying the same genetic locus. Bacteria may also express ABO phenotype. Springer characterized more than 100 strains and species that exhibit the ABO polymorphism. Springer is also famous for his chicken experiment. He grew up chickens under sterile conditions. The chicken did not develop anti-B antibodies, but when chickens were given diet contaminated with E. coli strain 086, which expressed B antigen, chicken developed anti-B antibodies. This is interpreted as what we call natural antibody. Actually not natural. They are the result of sensitization by the antigens present in diet or expressed on bacteria in the intestinal flora. A couple of ABO genes were later cloned, but thanks to the recent advancement of the sequence of the bacterial genome, we could retrieve many ABO bacteria genes and made a phylogenetic tree and color-coded the specificity. These genes were separated in two clusters, one representing A genes and the other representing B and AB genes. Bacteroides, such as E. coli, were found in two clusters, meaning that in addition to the vertical gene transfer, horizontal gene transfer occurred in the evolution of the bacterial genes. A and B antigen are the galactose linked by alpha-1,3 linkage at the non-reducing end of the sugar. But there are additional complex carbohydrates which have these sugars at the end. They are alpha-1,3 galactosyl epitope, Forstmann antigen, and IgB3. The last steps of the synthesis of these glycans are catalyzed by the enzyme named alpha-1,3 transferases. And the genes encoding these enzymes are evolutionarily related to the ABO gene, possibly resulted from gene duplications and divergence in specificity. A and B antigens, as well as other glycans, are genetically specified, but the expression may not be static. It may change during physiological conditions and also pathological conditions. There are four major categories of glycosylation changes in cancer. First, normal epithelial cells express AV antigens depending on the ABO status, but this expression is lost or decreased in cancer. And in some cancer types, this decrease is associated with prognosis, metastasis, recurrence, and survival. The antigenic loss rarely results from the gene loss, but rather the result of transcriptional depletion, which shows the hypermethylation of the promoter region of the ABO gene in cancer cells and tumors, which were decreased in the ABO expression. Group B or O individuals are not supposed to express the antigen. Group A or O individuals are not supposed to express the antigen. We humans are Forceman negative species and not supposed to express Forceman antigen because they are genetically incompatible. But in cancer, these genetically incompatible glycans may be expressed, although the frequency may not be very high. TF and TN glycans are responsible for microbial-associated TF and non-associated TN preaggregation of red blood cells. Together with cyto-TN antigen, these antigens are masked in normal tissues because of the additional sugar addition, but exposed in tumors. And this occurs very frequently, 70 to 90%. And this occurs in many epithelial cancer types and some of hematological malignancy. This is also the work by Springer. The fourth category is the enhanced expression Lewis blood group related antigen. Lewis X, Y, Lewis A, and X represent this category. And this enhanced expression is also observed in many cancer types. Actually, monoclonal antibodies against Lewis A and X have been used to monitor cancer recurrence by measuring the antigenic level in blood. In addition to the changes in expression, ABO has been associated with the incidence of ulcers and cancers. Group O individuals have 50% higher chance of getting ulcers. Group A individuals have 25% increased chance of getting cancers of stomach or pancreas. In 2009, the result of a genome-wide association study was published of the pancreatic cancer. The authors analyzed more than 500,000 SNPs in thousands of cancer patients and normal controls. They found 3 chromosomal regions with high association. The strongest association was observed with the SNP in the ABO gene, located in chromosome 9q34. In the old targeted approach, ABO association was examined. Therefore, there was a chance of introducing bias when selecting the LC corresponding controls. But in the case of modern genome-wide association studies, ABO association was found. Therefore, the results are more reliable and objective. Using GWAS, ABO gene has been associated with several diseases and many more biological parameters were associated with the SNPs in the ABO gene. For instance, the serum concentrations of von Willebrand factor and coagulation factor 8 are 25% lower in individuals with group O. And this lower concentration of coagulation factor 8 seems to be responsible for the lower incidence of venous thromboembolism, which is also called as economy class syndrome. Group O red blood cells infected with Plasmodium falciparum bind weaker to capillaries when compared with group A red blood cells infected with a parasite. And this lower binding seems to be responsible for the lower incidence of brain malaria in group O individuals. Contrastingly, one of Helicobacter pyloris adhesion receptors is RISB, and when functional A and B transferase are present, it added agarnac and galactose and modified RISB to LSB and BRISB, and bacteria binding less. Therefore, individuals have a higher chance of ulcers. In case of the pancreatic cancer, the molecular mechanism of ABO association has not been determined, and we are very interested in figuring it out. The disease association to ABO may be direct by the presence or absence of A or B antigens, as well as the presence or absence of natural antibody, but it may also be indirect through differential concentrations of metabolic and other parameters. Unfortunately, we are in the midst of COVID-19 pandemic. More than 150 million people were infected with SARS-CoV-2, and more than 3 million have died of COVID-19. During the past year, more than a dozen papers have been published reporting the ABO association with COVID-19 infection. Although there are a couple with negative results, the majority agreed a higher and lower risk of group A or O individuals. Genetic testing company 23andMe posted its own result by analyzing the gene sequence and survey of more than 750,000 participants. 750,000 participants. This is a very, very large number, and it should not be ignored. They observe the protective effect of phenotype. In June 2020, last year, the result of the genome-wide association study of severe COVID-19 with respiratory failure was published. In addition to a gene cluster on chromosome 3, ABO gene was associated. Many SNPs in the ABO shows a high association. SARS-CoV-2 virus is membrane encapsulated, so covered with membranes. Similar to other membrane encapsulated viruses such as HIV, measles virus, and SARS, the glycation patterns may resemble the patterns of the cells where the virus were produced. The binding of viral spike protein to angiotensin converting enzyme 2 expressed on human cells mediate SARS-CoV-2. Therefore, it is likely that the spike proteins may carry A and or B glycantigens depending on the ABO phenotype of the cells of where viruses were produced. If this is true, the virus produced in A phenotype cells may express A antigen. And this A virus can infect an individual or an individual without any inhibition by natural antibodies. But When infected to B or O individual because of the anti-A and anti-AB antibodies, this infection may be somewhat inhibited. But in case of the AB virus, it can infect to AB individual without problem, but to A individual, B individual, O individual, the infection may be somewhat inhibited. But for the O viruses, there is no inhibition by natural antibody. What it means is that not only the phenotypes, but also the combination and the directions are important for this inhibition. This is similar to ABO matched and ABO mismatched blood transfusion. It should be noted that the inhibition may not be 100% effective. In other words, a virus may infect all cells, but once infection is established, newly synthesized virus exhibit the phenotype of O, and it can infect other cells without any problem of natural antibodies. You can imagine that with this system, the inhibition is more effective in the ABO heterogeneous population than ABO homogeneous population. In Peru and Ecuador, more than 80% of the people have group O, but still these countries suffer from COVID-19 outbreaks, possibly because of this reason. In 2008, using the cellular model, anti-antibodies were shown to block the interaction between ACE2 protein and spike proteins carrying A antigen. But this experiment was performed using the spike proteins from SARS-CoV-2. And now several laboratories in the world, including my own, has been working to examine if this is the case with the SARS-CoV-2 spike protein. I already mentioned that group O individuals have 25 lower serum concentrations of von Willebrand factor and factor 8. And the majority of these proteins are synthesized in endothelial cells where A and B antigens can be expressed depending on the individual's ABO phenotype. Therefore, ABO polymorphism may directly affect infectivity and may indirectly affect severity. But these are just two examples of hypotheses, and there may be many other mechanisms. But it should be remembered that the odd ratio of severe COVID-19 in the respiratory failure is just 40%, 0.6 for group O individuals. If you compare this value with, for example, being older than 85 years of age, the odd ratio for hospitalization is 13 when compared with the people between 19 and 29, but the ratio raised to 95 when compared with people between 5 and 70 years. So the ABO effect may be very small. Therefore, it is wise to wash your hands frequently, wearing mask, keeping social distance, and getting the vaccination, whatever the ABO blood groups you may have. We are interested in the illustration of the molecular mechanism of the genetically incompatible glycan antigens. Postman antigens are expressed in some species, including mouse, but not expressed in some species, including humans. Postman antigens can be produced from the precursor globoside by the transfer of a GalNAc alpha-1 linkage by Forceman synthase encoded by GBGT1 gene. In 2012, my research team determined the cause of human GBGT1 gene deficiency. We prepared cDNA chimeras between mouse Forceman synthase and Forceman negative human GBGT1 gene encoded protein, and later made many amino acid substitution constructs. We used the same system, but with different cells, to examine the expression of Forceman using antibodies against Forceman antigen. We found two inactivating missense mutations in the human GBGT1 gene. We showed experimentally the reversion of one of the amino acid substitutions conferred human GBGT1 gene encoded protein with weak Forceman synthase activity, whereas the reversion of the two conferred strong Forceman synthase activity. However, these mutations were not found in the database of the somatic mutations in cancer. This shows the evolutionary tree of alpha-1,3 GalGanac transferases. The two amino acids corresponding to codons 266 and 268 are glycine and alanine, corresponding to the codon 266 and 266 of human A transferase are glycine and alanine in the GBGT1 gene encoded protein. In the majority of the GBGT1 gene species. But here we found mouse ABO gene. Although it is ABO gene, the encoded protein have these two amino acids. And we examined if this mouse ABO gene encodes the protein with Forceman synthase activity. The answer was yes. We also showed replacement of the codons 266 and 268 of human A transferase with glycine and alanine conferred weak Forceman synthase activity. In other words, in addition to GBGT1 gene, ABO gene can produce a protein with Forceman synthase activity. And because of this success, we searched for additional mechanisms that confer the Forceman synthase activity. And we found that methionine 69 substitution may also give weak Forceman synthase activity. We also showed that the exon 3 or 4 deletion, but not the exon 2 or 5 deletion of human A transferase messenger RNA conferred moderate Forceman synthase activity. Furthermore, the combination of the methionine 69 threonine substitution of the exon 3 or 4 deletion and glycine alanine substitution conferred human A transferase with strong Forceman synthase activity. We performed the same kind of experiment using FACS to obtain more quantitative data. And the result of immunocytochemistry were confirmed to be correct. To summarize this, we showed that human A transferase, which are encoded by ABO gene and not GBGT1 gene, possess intrinsic Forceman glycolipid synthase activity upon deletion of exon 3 or 4. Human red blood cells contain globoside, the precursor substrate of Forceman antigens. However, Boltzmann antigen are not synthesized in red blood cells because splicing is controlled under high stringency. But altered splicing is a hallmark of cancer. Therefore, we believe this explains some of the appearance of the Boltzmann antigen in cancer. We showed strong Boltzmann activity by introducing 69 amino acid substitution or deletion of exon 3 or 4 with glycine RNA substitution, possibly by the synergistic effect. The amino acid codon 69 is the first amino acid of exon 5. Therefore, 69 amino acid substitution and deletion of exon 3 and 4 seem to function in the same molecular mechanism, and we believe that may be due to the altered target localization. Now we are investigating that possibility. Additionally, we are studying the molecular mechanisms causing the expression of genetically incompatible A and B antigens in cancer. This ABO study was initiated when I was at the Biomembrane Institute in Seattle, Washington, in the USA. It started as a collaboration with Henrik Clausen, the head of the immunology lab, who isolated the human A transferase, and Seiichiro Hakumori was the scientific director of the institute. I'm very grateful to Manuel Pelcho, who invited me to join the MPPC. Laura now performs undergraduate final project in my laboratory. Maria Cristina did her master's. Emily has been with me for the past 10 years. His expertise in biochemistry and cell biology is indispensable to my research in the laboratory. And Miyako has been with me for over 25 years. Taking this opportunity, I would like to thank Joseph-san and Albert-san for allowing me to continue my scientific career at the Joseph Carreras Leukemia Research Institute. Miyako and I are very happy to be here. Thank you so much. I am also indebted to Evaristo, Manel, and Kiko for their generous support at the Institute. And finally, I would like to thank the audience for patient and kind attention. Thank you so much.
Thank you, Fumi, for this wonderful overview on the OVO genes and polymorphisms. So there are a few questions here. So I'm going to start by reading some of them. Okay. So the first one is, does a COVID virus produce in an endothelial cell present in its surface the ABA antigen?
That's what I assume, because that was demonstrated by SARS virus, not SARS-CoV-2. But because this protective effect of group O and other etiological data suggests that there are some inhibitory mechanisms in all individuals. And I think that because of the natural antibodies, anti-A and anti-AB and anti-B in all individuals, meaning that the antigens should be present. But I'm pretty sure.
So there is a second question regarding this one that says, if yes, as you said, a B blood type individual? If yes, okay, a B blood type individual gets infected with COVID produced in an A blood type person, would the immune system of the B individual neutralize the virus more efficiently with its anti-B antibodies?
So for the quantitative effect, that has not shown, demonstrated experimentally. So we don't know, but most of the anti-A and anti-B antibodies are IgM, and that stayed inside the blood. And the antibodies working to inhibit the infection should be IgG or IgA. And then in that case, group O individual have anti-AB, that is of IgG nature. But for between the comparison with anti-B or anti-A, there are a couple of papers suggesting that anti-A works better than anti-B. But compared with anti-AB in or group individuals, that may be much weaker.
Thank you. I have another question regarding COVID. So does the expression of the antigens, in this case A, changes with age? And this could explain the infectivity of the virus? In older people, like higher infectivity compared to young people?
Yes, yeah. The expression of A and B antigen decrease while we age. Okay, and that may explain also the concentration of von Willebrand factor, factor 8, those increase by age, and I think many factors are involved. And then with the experimental system, we can demonstrate if that is theoretically possible or not. But in the case of infection, you can see in the timeline, some countries suffered a first wave and then nothing. But because of some measures taken by the government, and everything changes. So even if it doesn't fit to the theoretical basis, that doesn't mean that this is not happening.
Okay, I see. Thank you. So we have another question from the audience that says, okay, I need my glasses. What are the prospects of using glycosylation pattern changes during disease as the biological basis for possible future therapies?
Yes, that is what I have proposed several times. Because we are interested in the cryptic glycantigens or genetically incompatible glycantigens, is because we can create novel cancer immunotherapy targeting those antigens. And there are many possible ways, but we plan to utilize red blood cells exhibiting these antigens as a means of immunize to enhance the immunity against these antigens.
Okay, thank you. So another question from the audience that says, the prevalence of cryptic lichen antigens seems to be very high in individuals with cancer. Can that affect the efficiency of the therapy?
So the immunity, both cellular and humoral immunity against cryptic glycoantigens are present because for example, TF antibody is a major portion of IgM antibodies. But the fact that cancer grows shows that the immunity is not strong enough to suppress the progression of cancer. But by enhancing the immunity, you may suppress, not only predict or I think we can make the new therapy based on the biological or basic findings based on this phenomenon.
Okay. So more questions. So apart from the transfusion matching, what other functions have A/B antigens?
Functions. As you said, transfusion and transplantations are medical protocols produced by humans. So those are not the intended purpose of polymorphism. But for example, these antigens are exposed on the cell surface, meaning that they can interact with viruses and bacteria and also other cells. And then the modification of A or B or G's should affect the interaction. And also that may affect the interaction between the receptors and ligands also.
Okay.
But because the fact that they are present as polymorphism means that it's not indispensable. But this variety may give the advantage for the survival of the species rather than survival of the individuals.
Okay. Thank you. So we have time for one final question, and this is, would you recommend to monitor a specific cancer disease development depending on the ABO polymorphism in a personalized manner? Do you want me to read it again?
Yeah, could you?
Yeah. So would you recommend to monitor a specific cancer or disease depending on the ABO polymorphism in a personalized manner?
So you mean because of the higher rate of cancers of stomach or pancreas for individual?
Yeah, I guess what the question means is whether this is something we have to take into account for personalized medicine in disease settings.
Yes,
Yes and no.
But for me, whichever the ABO phenotype, the treatment option should be quite similar because this glycation may affect biologically, but always this kind of effect is not 100% or zero, but it change a little bit.
Okay.
So
yeah, I mean, depends on the disease, I guess, too.
Yes, for some other diseases, there may be no correlation, so you don't have to worry about that. But even with those with association, the treatment option may be difficult to develop according to the ABO phenotype.
So maybe I finish with a final question for myself, in which I think you have nicely shown studies in which they prove the correlation of the ABO with specific cancers. But I think I cannot avoid asking you whether these cancers also involve the hematopoietic malignancies, because you put a lot of so you have provided examples of solid cancers, but you have not said anything about hematopoietic malignancies.
For ABO, yes.
ABO.
But the expression of A or B antigen decrease in the leukemic patient because these cells are released prematurely to the bloodstream from bone marrow. Okay, but that's all they are not maturated. But how can we so that is a very important question working at the research institute specialized in leukemia. In science and medicine, all are related. The hematopoietic stem cell transplantation was used to monitor the cell fate of hematopoietic stem cells, but now it is used for leukemia treatment. Blood transfusion is used to ease the anemic symptom of leukemic patient. We clone the ABO allelic cDNAs from the cancer cells of stomach and colon and illustrate the molecular genetic basis of the ABO blood group system without using any red blood cells. So we may make contributions in leukemia research so far as we keep in mind the mission of the Institute to cure leukemia and share the passion with Mr. Joseph Carreras. So for the ABO, you may not see the direct association or direct relation with the leukemia, but actually blood transfusion helps many.
Exactly. Okay, Dr. Yamamoto, many thanks for this wonderful talk. I think it was a success based on the amount of questions that you got. So thank you very much and enjoy your afternoon.
Thank you so much, and thank you for the audience.